We are talking about mechanism of muscle contraction, means how the muscles contract. It is about mechanism of muscle contraction. See, muscle contraction, if it is voluntary, muscle contraction, it means it is in your control. So that will be taken care by the help of skeletal muscles, the muscles which are attached to your bones. For example, if I'm writing, so I'm writing with my fingers and hands, so the muscles are helping me to write. So here, before starting with this, we need to understand little bit about structure of skeletal muscle fiber. We are going to talk about structure of skeletal muscle fiber, how exactly it looks. So for this, I will be drawing one diagram. So normally when I look at this structure, it appears something like this, where in between it is going to show striations. Because this is striated one, it is voluntary in nature, it is in our control. So when I look at this, what I can observe, this area is called as dark band, and this area is called as light band. So when you observe very carefully, you can see alternate dark and light bands present. This area is again a dark band, light band, dark band, light band, dark band. What is the other name for dark band? Dark band is called as A band, means an isotropic band. It will not allow the light to pass through it. So it is called as an isotropic band. Whereas the light band is called as I band. I band means isotropic band. Isotropic band means it will allow the light to pass through it. So when we talk about the skeletal muscles, I can say that these skeletal muscles fibers, they are long. Most important, they are all unbranched. The covering that you see basically is called as sarcolemma. That is the covering present. And this cytoplasm is called as sarcoplasm. So what is sarcolemma? It is nothing but the covering. And what is sarcoplasm? It is nothing but the cytoplasm of the connective tissue. Now, most important, we need to remember this. They have something called as myofibrils. This becomes the very important part. When you talk about myofibrils, there are two types of myofibrils that you will study. One is primary one, that is helping in contraction, and other one is basically secondary one. So we have primary and we have secondary. The primary one is called as myosin and the secondary one is called as actin. So remember, actin and myosin are the two important muscle proteins for contraction. The actin is basically attached to Z line. Now the point is, what is Z line? So you have to remember, in the light band there will be a dark line present. So this dark line that you can see, it is called as Z line. So we can say here that in the light band there is a dark line present. That dark line is called as Z line, and the distance between the two Z line is called as sarcomere. And remember, sarcomere is the unit of contraction. Now let's see this structure. We are going to focus only on this structure in detail, only one light band and a dark band. So for that, we have to draw a line. So let us take it in this way. So we draw this as the line. You can also call it as sarcolemma covering, and then we draw this line. Now here we are going to draw the most important part. So I take it in this way. There is a line like this. This is one band, Then we have this other one. So this is the another one. We take one more band here. So I take it in this way. So this is also a band. So let's take it in this way. This is a band here. We take it here also. So basically, we are drawing the diagram for the bands that we normally study. So this is also a band. So draw it from here. This is also a band. And I take this. This is the important diagram. And after this, we are going to draw a big one. Where is this one? So now this is important. You have to take it from here. So observe these lines now. These are very important lines. So here this pink colored one that we observe, this is called as actin. Actin is basically, you can say, thin and attached. This is actin, the pink color one. When you look at this, the green color one, it is called as myosin. So when we talk about myosin, remember it is thick and it is free. It means it is not attached to anything. Let's understand here this area, we call it as light band. So only this much from here till here, I can call it as I band. That is isotropic band. It will allow the light to pass through it. And from here, from this part, from where the green part starts till the another green part, this is called as A band. That is an anisotropic band. Light will not allow to pass. And again, from here till here, you will get the I band. So I band is called as light band and A band is called as dark band. So we have light band, dark band, and this way present. In the light band, there is a dark line present. So we draw this dark line. So this dark line that you can observe here, it is called as Z line. So in the I band, you see a dark line present. So this dark line is called as Z line. So same Z line will be present here also. So there is a Z line present here. So I draw this. So now when I do the labeling, this line is called as Z line. This is also Z line. And distance between two Z line is called as sarcomere. So I can put it in this way. So let's take this and this to be Z line. So distance between the two Z line is called as sarcomere. What is sarcomere? Basically, I can say sarcomere is nothing but contractile unit of muscle. Or even I can say sarcomere is unit of contraction. The muscles, they will contract. So it is possible only because of sarcomere. In the light band, there is a dark line. But at the same time, in the dark band, there is a light line present. So this band that you observe here, this much area, this area that you observe in between, so this area is called as H band. So remember, in the dark band, there is a light line present. That light line is called as H band. There is a connection now. There is an M line. Because this myosin is thick and free basically. So the fiber which binds the M or the myosin, so this is called as M line. So this blue line that you observe, it is called as M line. So logically, what is M line? You can say it connects and binds sarcomere. So the one which connects and binds sarcomere, I can call it as M line. This is the straight view. When I take a cross section of this, if I have to cut this and I have to observe how it will appear. In the cross section, I will see that there is one green color one. So this green is nothing but, I can call it as myosin. The myosin is surrounded by actin. So here there will be 6 actin present. So you can say 1, 2, 3, 4, 5 and 6. So remember, for every one myosin there are total 6 actin present. So I can say for every one myosin you will get 6 actin. Now for example, if I draw this as the myosin, so already there are 2 actin present here, 1 and 2. So I will say 3, 4, 5 and 6. Like that it will be 6. Now if I draw one myosin like this, so here you can see 1, 2 and 3 already present. So we draw 4, 5 and 6. So like this you need to remember, for every, you can say 6 actin, there is one myosin. And one actin is surrounded by 3 myosin. One actin. So if I talk about this actin, let us take this. This is one actin. This one actin is surrounded by 3 myosin. You can see 1, 2 and 3. So I can say for every one actin there are 3 myosin, and for one myosin there will be 6 actin. So let's take this. This is one myosin and there are 6 actin. 1, 2, 3, 4, 5 and 6. This is how the arrangement is there. There is a very important tubule called as T tubule. Now this T tubule is present here. This tubule that you can see here, this is called as T tubule. And T tubule is the most important one because it is responsible for spreading the impulse. So I can say this is T tubule, and the T tubule role is, it helps in spreading of impulse. So if the impulse is conducted, it is conducted because of T tubule. Now we have to study this, how actin and myosin look. That is the important part. So let's look at the structure of actin and myosin. So when we talk about first structure, so let's put the heading as structure of actin and myosin. Once we know the structure of actin and myosin, most of the things will be solved very easily. So let's go with respect to the structure. Very first, I'm going to talk about myosin. So the myosin structure, I will say that the myosin exists in two forms. One is the polymer and second is the monomer. So polymer is called as myosin. This is a polymer. And monomer is, polymer is myosin, but the moment I talk about monomer, the monomer is not called as myosin. The monomer is called as meromyosin. When I say meromyosin, this meromyosin, it consists of two parts, the head and the tail. So let's understand with the diagram. So I draw this two as the head. So this is the head of myosin and there will be tail. So I draw this as the tail. There are two tail. So both are intercoiled with each other. So like this. On the head, you will find some special receptors present here. So I put it in this way. There are two important parts with respect to the head of the myosin. So I put it in this way. So let's understand, this is one meromyosin. So very first thing, this part is nothing but it is the head of the myosin. This lower part, it is nothing but the tail of the myosin. The head is called as HMM. What is full form of HMM? It is called as heavy meromyosin. And the tail is called as LMM. So what is the full form of LMM? It is called as light meromyosin. So remember one thing, that heavy meromyosin and light meromyosin, is with respect to the molecular weight. The head has high molecular weight, so it is called as heavy. The tail has low molecular weight, so it is called as light. There are two binding sites present on the head. So one is called as actin binding site. So this I can call it as actin binding site. And the other one that we have is ATP binding site. So there are two binding sites present on the meromyosin, actin binding site and ATP binding site. This is one structure that you have observed. But when I put it in bundle, it will be totally different identity. For example, if it is like this, these are all the threads, you can say, of the meromyosin, the tail. And finally here there will be head present. So like this, you can see there will be two head present. So these two head, they are indicating the heavy meromyosin. So here you can understand that there are many myosin present together and they are winded around each other. So this is how the structure look alike. If I have to draw it in a simplest way, I can put it in this way. How the bundle of meromyosin will look alike. So it will appear something like this. Here there will be no doubt all these are the tail. So I put the tail here as such. For head I will be using different colors so that it becomes very easy for you to understand. So this is the head. So there will be many head as such present and they will be forming a bundle. So this two, what you can see, this is nothing but the head. There are two head present, so we put it in this way. So this is how a polymer of myosin will appear. It will have many head present and tails, and all the tails are winded to each other to form a fiber-like structure. So this is how these things will appear. And now this is the complex form of the myosin. So remember, all these are attached to each other at a point. So this point where they are attached here, this is very important line. This is called as M line. This is called as M line. And M line means myomesin line. So all these tails, they will converge at one. That is called as M line. This is all about myosin. Let's understand the structure of actin. So whenever we talk about actin, remember it is also in the form of polymer and monomer. So actin is nothing but, it is called as secondary contractile protein. So this secondary contractile protein is called as actin. Now it also exists in two forms. One form is in form of polymer, and second you can see actin in the form of monomer. When we talk about polymer, in polymer I can say there is F actin. Polymer is called as F actin, where F stands for fibrous. So it is also known as fibrous actin. When we talk about monomer, monomer includes G actin. So what is G actin? It is called as globular actin. So in actin also there are two, the polymer and the monomer. The polymer is called as fibrous actin and the monomer is called as globular actin. If I have to draw the diagram of these structures, that is very important to remember how exactly the actin represents. So remember, actin basically consists of tropomyosin, troponin, and the G actin or the protein. So how you will draw it in structure? So I put it in this way. Let us take this. This is the complex form. So I take it here. Let's make it a little bit thick so that the line remains visible. And after that, we are going to draw this. These are all the proteins that are present surrounding this. So you have this as a structure. This is how the actin is going to appear, and troponin will be there. So I take it in this way and we draw this line. This is very important line because this is that fiber which is binding all the troponin and the tropomyosin. So we got this. Now on this you will have some special protein present that's called as G actin. So I put it this way. This is nothing but the G actin. Let's understand now. Let's do the labeling of this so that we can understand what we are trying to say. This, what you're observing, the black color one, it is called as tropomyosin. The component that is like this is tropomyosin. At interval, you will have troponin. So this blue color structure that you're observing, it is present at intervals. It is not present regularly. And this structure that you can observe here, it is present at an interval. So this structure that you can see at an interval, it is called as troponin. So remember, tropomyosin at intervals has troponin present. And this pink colored one that you can see is nothing but it is the actin protein. So this is called as actin protein. On this actin protein, I can call it as G actin, that is nothing but globular actin. So like this, you will get the entire structure of actin. Now to understand this, we have one very important statement. It says that the troponin plus tropomyosin, troponin and plus tropomyosin, what they do, they together block active site at rest. So if the muscles are at rest, there is no conduction of impulse. So I can say that these troponin plus the tropomyosin, they will block. And once the active site is blocked, it means there will be no muscle contraction taking place. Let us understand how things are there. So I take this red color structure. This red color structure, I call it as actin. Now there is troponin involved. So this blue color structure, I told you troponin. So this part is the troponin. Troponin has basically blocked this. This is the active site. So the active site is blocked because of troponin. So if I draw this, the structure will appear something like this. So I can say that the active site is blocked by the help of troponin. And this is the tropomyosin basically that will connect the two troponin. So when I do the labeling, how it will appear, this entire stuff is actin. This is the troponin, and this site where it is blocked, it is called as active site. So I can say when there is no muscle contraction, the active site is blocked because of this troponin. So this is at rest condition. But when the muscles are working, then what will happen? What will happen at the myosin head? So if this is at rest, the myosin head will be normally free. So I draw this way. So this is the myosin head, and this myosin head is free. So I can say at the resting stage, at rest, myosin head is free. And since myosin head is free, it cannot bind to the active site also. Why it cannot bind to the active site? Because the actin is blocked. Actin is blocked by troponin. Somehow, if I can remove the troponin, then the head can easily bind to that active site. Most important part is for any muscle contraction, we require energy. So let's understand first how the energy is obtained. So we are going to talk about the next important part, that is the energy for muscle contraction. How the muscle contracts? that is different story, but how the energy is provided, that is we need to know. Energy for muscles contraction. So we have two options: vertebrates, and in few invertebrates you see this kind of mechanism for getting energy. Vertebrates and few invertebrates, you will get this method for getting energy. So what are the different sources of energy? So remember, the energy or the ATP is provided by the help of special chemical, and that chemical is called as phosphagens. Phosphagens are those chemicals which will provide energy. One is in the form of ATP, direct source of energy. Second is phosphocreatine. It is also going to provide energy. So phosphocreatine, or even if you want, you can call it as creatine phosphate. What are phosphagens basically? So we can say phosphagens are nothing but these are energy providing compounds, the compounds that will provide energy for us. For example, if you are running, so initially you will use all the ATP as a source of energy. But what if the ATP gets over and still you continue to run? Under that condition, how energy is provided? That is what this entire topic is all about. So here we put the very first situation. The first step is ATP. So when you are getting energy in the form of ATP, this ATP will be first broken down into ADP and IP. What is IP? It is inorganic phosphate. And finally energy will be released. When one ATP is broken down, 7.3 kilocalorie of energy is released. So this energy, which is released by the breakdown of ATP, is used for actin myosin cross bridge formation. Remember, this energy is used for actin myosin cross bridge formation. So this actin myosin cross bridge formation will help in the contraction. And here you require one enzyme, and the enzyme is called as ATPase enzyme. And this ATPase enzyme is present where? It is present in the myosin head. Most important. So therefore, this is how the energy will be generated. But what if the energy is less? Because we all know that since number of ATP in cell is limited, so under such condition, if you want more energy, then how things will work? So we have to go for step number 2 for getting ATP. The step number 2 says generation of ATP. Now we need to generate ATP. Definitely ATP will not be generated that much, but still some amount of ATP will be generated that will help you to carry on your process. So here we have a creatine phosphate. As I told you, we get creatine phosphate also that is present in our body. So this creatine phosphate will combine with the ADP, that is adenosine diphosphate. And in the presence of enzyme, it will combine to form ATP plus creatine will be released. So this is how you get more ATP. But this is taking place in the presence of enzyme, and the name of the enzyme is creatine kinase. This will provide additional amount of energy. But again point comes, what if more ATP is required? If more ATP is required, then under such condition what we can do? So if more ATP is required, then the body will search for third source of energy, because ultimately body will never give up. It's like a lion running behind you. The lion is trying to chase you and you will run to your maximum. You get tired, still you will keep on running. So for that run, you need energy. That energy is provided by generation of ATP by the help of creatine phosphate. But if this also gets over, then what else? Then there is anaerobic breakdown. Remember, anaerobic respiration, because see, when we run and we don't breathe, what will happen? All the glucose will be converted into pyruvic acid, but that pyruvic acid will be converted into lactic acid. So under anaerobic breakdown, what will happen? I can say that the glycogen will be broken down to give you glucose, and this glucose will follow anaerobic respiration and it will form lactic acid plus energy will be released. But here remember, due to anaerobic respiration, the energy released will be very less. So I can say energy released is less, but still it is used for running, and this energy will be used for doing the work. And the next is, what if you need more energy? What happens to the most of the lactic acid? So remember, most of the lactic acid goes in liver. Most of the lactic acid goes in liver. And what exactly happens there? So the lactic acid which has reached the liver, it will be divided into two parts. So I can say fourth, fifth part basically. So I can say 4 by 5 part will travel in the liver, but 1 by 5 part will not go in the liver. So fourth, fifth part will be converted into glycogen again, and this glycogen will be then stored. But 1/5 part will be again broken down. So once it is broken down, it will give you energy. So 1/5 part is broken down to give you what? Carbon dioxide plus water plus 2 ATP. Again the ATP is less energy, but still some ATP is generated, and this ATP is then finally used. So these are the different sources by which your body can get energy. And how to generate the creatine phosphagen again? So you have to use simple step. Creatine, when you eat food, the food will give you energy. So creatine combines with that energy, and finally it forms creatine phosphagen. So this is the way how energy is provided. Now let's understand the mechanism of muscle contraction step by step. So we are talking about now mechanism, how the muscles contract. So mechanism of muscle contraction, how the muscles contract step by step. So first thing that is required is creation of the impulse. And you need to remember one more thing, that there are two types of muscle contraction basically. One, I can say it is going by electrical, so there is electrical event, and there is one more that is called as biochemical event. So you have two types of event, electrical event and biochemical event. So this electrical and biochemical event was given by one of the scientists in the name of Albert Zent. So he was a Japanese scientist basically who talked about this. So it is Albert Zent. And what he mentioned in this step. So let's start with the first step, that is called as step number 1. What happens in the muscle contraction? So let me first draw a diagram for you that will help you to understand how things are going. So we draw this. This is the covering called as sarcolemma. Then we draw a Z line. And if you remember, this was the Z line, the unit of contraction. So this is the Z line. Then we draw the actin filament. So basically actin was like this. So I draw this as the actin filament. Now we draw the myosin. So we take this. Now myosin I will not be making complete. So I will be showing only the head and the myosin fibrils. So I take this as the myosin. Now in this myosin there will be definitely head present. So I take these two as the head. But initially the head will be not bound to the actin because the active site of actin is blocked. And there is one more, the one that is responsible for the impulse. So this is nothing but T-tubule that will generate the impulse. When there is no conduction of impulse, so outside will be positive charge and inside will be negative charge. So let's do the labeling first and then we will understand. This is called as sarcolemma, the outermost covering. The red one is called as actin filament. This is called as myosin filament. This blue one is called as Z line. The unit of contraction is the distance between the two Z lines. So we have Z line. This yellow one is called as T tubule, that is responsible for conduction of impulse, and the myosin filament. So what is this condition called as? This is called as resting membrane. When I use this word resting membrane, it means what? The cell is actually resting. It is not doing any kind of activity. So what we have to understand with respect to resting membrane? Very first, that this condition is nothing but a polarized state where there is more electropositive outside and electronegative inside. So we have more of electropositive outside and electronegative inside. And after this, what is the condition here? It is somewhere around minus 60 millivolt. This is the resting membrane. Now, once the impulse is conducted, then what will happen? So this is step one. Let's talk about now step two. So when we talk about step two, how the things will work. So let us talk about step two. So in step two, I can say it is a depolarized kind of impulse. So again, let us draw that. So we draw this as the sarcolemma. Then I draw this as the Z line, that is the dark line that is seen in the light band, that is called a Z line. To this we will draw actin. So I draw this as the actin. And then we draw the myosin. So let us take this as the myosin. So I take this as the myosin. And this there will be a T tubule that is responsible for conduction of impulse. This is the active side which is blocked by the troponin, and this is the head of the myosin. Now under this condition, see, when the impulse is conducted, what is this? This is nothing but the axon ending. So I can call this as axon end bulb. Let's call it as axon end bulb. And this axon end bulb will be filled up by some chemical, if you remember, that was called as neurotransmitters. So this neurotransmitter now will conduct the impulse. So when it goes in, it travels into the T tubule. So from here it will spread. And once it is charged, what happens? The outer side becomes negatively charged and the inner side becomes positively charged. The neurotransmitter that is generally seen in muscle contraction is acetylcholine. And this is T tubule. Now how the things are going to work, just try to understand. Active site of the actin is blocked. This is the actin. So the actin active site is blocked because of troponin and tropomyosin. So I can say, and since it is blocked, the myosin head, basically, the myosin head cannot bind to the actin because it is blocked. But due to conduction of impulse, what has happened? We can say now this membrane is a depolarized membrane. So I call it as depolarized. Depolarized membrane means what? When the impulse basically is conducted. So I can say in depolarized state, action potential is generated. Action potential is generated. And since action potential is generated, the sodium pumps will open up. And finally what happens? That the axonic bulb, it binds to sarcolemma. Sarcolemma is the outermost covering of connective tissue. Once it has binded, what will happen? The permeability to membrane will increase. And since the permeability to membrane increases, outside will become more electronegative and inside will become electropositive. And once this is done, the most important thing, that tubule is responsible for release of calcium ion. So your muscles cannot contract if there is no calcium ion released. So there is a release of calcium ion from sarcoplasmic reticulum. So what we need to understand here is that the impulse, when it travels through this T-tubule result in generation or activation, so that what is released? More of calcium ion. And most important point here is that the active site of actin is blocked. Active site of actin is blocked. And this active site of actin is blocked by troponin and tropomyosin. Remember one thing here, there is a love story going on. That the troponin, even though it is attached to actin, but troponin loves calcium. So due to conduction of impulse, when the calcium is released, what will happen to the troponin? The troponin will leave the active site of actin and it will go and bind to the calcium ion. So that becomes our step number 3. So let's look at step 3. What goes on in step 3? So very first, let us draw the diagram. So this is nothing but the sarcolemma, the outermost covering. Then there is a Z line present, so I can call it this as the Z line. Then there is T tubule, so I call this as the T tubule. Let's draw the actin. So we have the actin. Then we draw the myosin, so I draw this as the myosin. What is going to happen here? So remember, this is the active site of actin which is blocked by troponin. So this blue colored stuff which has blocked the active site, so I can call it as troponin. Troponin has blocked the active site. So this active site now has to be released. This is nothing but the head of the myosin. So leave it as it is. Now since there is lots of calcium ion present because of the conduction of impulse, there is lots of calcium ion present in the cytoplasm. So let us take this green color stuff as the calcium ion. So this is all calcium ion. Now this calcium ion is present in the cytoplasm and it is moving here and there. Looking at the cytoplasm, what will happen? Looking at the cytoplasm, this troponin will be released. The troponin will leave the active site and it will come and bind to the calcium ion. So let's take it in this way. This will be released, it will come and bind to the calcium ion. Here what I can say, that the troponin, I can say troponin leaves the actin. Troponin leaves the actin and binds to calcium ion. This is very important part. Once the calcium ion has taken up the binding, so what will happen now? This actin is now free because all the calcium ion has been released from there. So I can say now the active site is free. So here you can see that this active site is now free. The calcium ion, the troponin is released and it has bound to this. So here I can say that the active site is now free. So I can say that active site is free. If there is no calcium, remember there will be no contraction of the muscles taking place. Now what will happen? The myosin head will bind. So what we need to understand here? We have to say that the troponin leaves the active site leaves the active site of actin and binds to calcium ion. Therefore, the active site is free. Now, since the active site is free, what will happen? The myosin head binds to the myosin head binds to active site. This is the very important step taking place in cross bridge formation. So how it will appear? Now to show it how it will appear, we will show in this way. There is sarcolemma. Then we draw this as the Z line. Now to this Z line there will be T tubule. We draw the actin. So we draw the actin here. Then we draw the myosin. So the active site is now free. Now since the active site is free, what happens? The myosin head binds to the active site. This is the important event. Why it has binded to the active site? Because the troponin is free moving with the calcium, because troponin loves calcium, not the active site. So since this calcium ion has kept the troponin busy, by the time what happens, the myosin head basically binds to the active site of the actin. So this is now actin, this is myosin, and this is a cross bridge formation. So this entire setup we can call it as actin myosin cross bridge. Remember, so it is called as actin myosin cross bridge. The next step, now this is the third step which has taken place finally, and this actin and myosin cross bridge formation, it will result now hydrolysis of ATP. So as it is bound, now one ATP will be hydrolyzed. So after this what happens? One ATP is hydrolyzed. Why? Because on the head there is ATPase enzyme and that will break. So one ATP is hydrolyzed. Now since one ATP is hydrolyzed, there is a formation of cross bridge. So I can say cross bridge formation takes place, and this cross bridge formation taking place, it is called as, now it will lead to power stroke. What is power stroke? This head will try to pull the actin towards inside. So let me draw one basically power stroke. So I take this, then we take this. So what exactly will happen here? The structure is now going to bend. Look here, it was straight, but now it has pulled. Basically what will happen? The structure will come this way. So this is called as power stroke. It means it has pushed the actin in one direction, but now the head cannot go further, so it cannot take it much further, so it leaves it. So it will go for another step, that is called as recovery stroke. So what will happen in the recovery stroke? Basically the actin, now you see, because of this power stroke, what happens? The actin moves inward, but once the power stroke is done, the actin, now the head will be removed. So now the myosin head moves back. So how it will appear? So with respect to structure, I can say there is a recovery stroke. So let us take this and let us take this. So when you look at the position, you will understand. Now this is the head here. Now it has not binded to anywhere, and this is the active site. Now since it is the active site here, what will happen? The other head, because as I told you, in the polymer there will be many head present. Now this head will bind and will try to pull it here. So like this, there is a sliding filaments taking place. So this theory is also called as sliding filament theory. And because of this, most important, the contraction of the muscles takes place. Since there is taking place in both the direction, I can say that the sarcomere basically contracts. So this power stroke and recovery stroke helps the muscles to contract. So let's understand again by the help of separate mechanism that is called as sliding filament theory. This is the next one that we are going to study. So we are talking about sliding filament theory. So what will happen in this filament theory? What this theory results in muscle contraction. The scientist who talked about sliding filament theory is A.F. Huxley. Even there is H.E. Huxley, Jensen, many other scientists are involved. So what is the first step for sliding filament theory? The first step is it receives a stimulus. The muscles, it receives stimulus. And after receiving stimulus, what happens? The impulse travels till the neuromuscular junction. And the neurotransmitter will be released. What is that neurotransmitter? It is acetylcholine. And this neurotransmitter will activate the T tubules. And when the T tubules are activated, what will happen? The calcium ion is released. So this is the very first and the simplest step. So I take this as the sarcolemma and I take this as the neuromuscular junction. What will happen? This is sarcolemma. Sarcolemma is nothing but the outermost covering. What is this? This is axon ending. And finally, in between, there will be chemical. So this is called as acetylcholine. And this will travel in the T tubule that will result in the generation of more of calcium. So what is step number 2 now? Step 2 is the release of calcium ion. So let's take it in this way. Step 2, the calcium ion will be released. So once the calcium ion is released, step 2 is calcium ion is released. And as I told you that the troponin is more attracted towards calcium. So calcium ion is released from where? From T tubule. And once it is released, what will happen? Troponin leaves the active site. Troponin leaves the active site of actin and binds to calcium ion. And once it binds to the calcium ion, what happens? The active site is now free. And once the active site is free, what will happen? Step number 3. What will happen in step 3? We can say that now troponin and tropomyosin is removed. As a result, what happens? Active site gets exposed. Once the active site gets exposed, the myosin head binds to the active site. Myosin head binds to the active site. And this binding of the myosin head to the active site, it is going to result in the formation of step 4. What is that step 4? Actin-myosin cross-bridge formation. It is called as actin-myosin cross-bridge formation. So this is very important part that we have to remember. So there are many head binding and taking place. So I can put it in the diagrammatic form. So just to make you things clear. So let us take this to be the myosin. To this there will be head. So I take this as the head. There will be many head, but we are drawing only two. And then we draw the actin. So this is the actin. Now to this actin, what is there? There is troponin attached. So looking at the calcium ion, so there is a calcium ion here. So this calcium ion will attract. It will attract the troponin, and finally the troponin will leave this active site. It will go and bind to the calcium ion. Once it has binded to the calcium ion, how the actin will appear? So I can say the actin will appear like this, where the active site is now free. There is nothing present here, so the active site is free. And what will happen after this? The head will bind to it, and once head is binding, it is going to pull it. So let us take here, the head will come and it will bind to the active site. Very important point. Unless and until there is a calcium ion, the muscles cannot contract. So now this is what is actin myosin cross bridge formation. This is actin, this is myosin, and this is cross bridge. So this actin and myosin cross bridge formation takes place, and this head will move in this direction. As a result, this entire actin will be pulled inward. So I can say now the actin is pulled inward, and once it is pulled inward, there will be a recovery stroke. See, this pulling is basically because of power stroke, where the ATP gets hydrolyzed and it is broken down. The power stroke will result in pulling it back, pulling it towards inside. Now the last step comes for muscle contraction, that is step number 5. This is sliding filament theory. Step 5, myosin head moves backward. Now it will leave. So I can say myosin head moves backward. And since it is moving backward, what will happen? There is a stroke called as recovery stroke. And this recovery stroke results in what? The Z line comes closer. Very important point. The Z line, it comes closer. Sarcomere contracts. And since the sarcomere contracts, I can say the muscles contracts. So this is how the muscle contraction takes place. So there are certain important elements that we should all remember and we should never forget. Very first is the structure of actin. Second, you should know the structure of myosin. Third, you should talk about the neuromuscular junction. Fourth, you should talk about T-tubule. Then you will talk about calcium ion, which is very much essential for contraction. Then you talk about troponin. Troponin leaves. After that, you will talk about actin myosin cross bridge. Then 8th point, you will talk about the power stroke. And once power stroke is over, the last stroke will be recovery stroke. So, and once the recovery stroke is done, what happens? We talk about the 10th point, that Z line comes closer. This is how the contraction of the muscles takes place.